Good morning. Can you all hear me?
Yes, yeah, loud and clear.
Okay, thank you, Professor Dowd. And it's great to be doing this webinar on behalf of the Biochemical Society and Portland Press. So before I begin, let me introduce what the lab does generally. We're looking for developing therapeutics for chronic pain and addiction. And we do this by examining new targets and specifically ion channels and their regulators. Some of the targets that we've been working on are, for example, the N-type calcium channel, which is a very well documented target for chronic pain. We've discovered new targets, which I'm going to talk to you about today, particularly cytosolic proteins that are regulators of key ion channels and pathways involved in plasticity of pain. And maybe I'll have a little bit of time to talk about this really exciting story we found by discovery of a new molecule called neuropilin, which acts to induce analgesia through the spike protein. We also are fortunate to be working at the University of Arizona in collaboration with physician scientists working on non-pharmacological development of management of pain, as well as non-opioid based products from natural sources. So why are we doing all of this? Well, it's because chronic pain is a massively complicated condition. It spares no part of your body and mind. And despite the fact that chronic pain patients are often waiting about 10 years for a diagnosis, only a few of them are able to get, only a fraction are able to get adequate pain management. And it's for this reason, because pain is complicated, as I said earlier, it can be both governed and by mechanisms that originate in the central and the peripheral nervous systems. There is support for the idea that the peripheral nervous system itself may be important and sufficient to drive neuropathic pain. Particularly in recent work, there has been evidence suggesting that hyperexcitability driven by the nociceptors in the dorsal root ganglia are sufficient to drive this neuropathic pain. And particularly that is done by ion channels. In the example I'll give today is a voltage-gated sodium channel that is housed at key sites within the dorsal root ganglia, as well as in the nerves, both at the periphery and in some of the pathways here at key sites within the pain circuitry. And a recent paper suggests that this may be the sort of the prototypical low-hanging fruit, if you will, and could lead to the development of successful therapeutic compounds for managing pain. So let me introduce this main player. Sodium channel 1.7 is a key channel. It defines the threshold of the action potential. It also contributes to the upstroke of the action potential. Really, the efforts to study this channel were galvanized by the discovery that mutations in the 1.7 channel cause human pain syndromes. And this is really what led people to go after this channel as a potential target for pain. It's present in the right locations, in the skin, in the periphery, in the dorsal root ganglion neuron, the nociceptive neurons, as well as in lamina 1-2 of the dorsal horn of the spinal cord. And so we've been studying this for a while, and our efforts really were gravitating towards finding regulators of the trafficking and activity driving this particular channel. And about a decade ago, we discovered a protein called CRIM2, collapse in response mediator protein, which is really an axonal guidance sort of protein. And for the first time, we discovered it to associate with sodium channel. And then that embarked a series of studies which led us to show that if you take out from heterologous cells or from sensory neurons the expression of CRIM2, you saw a reduction in the sodium currents, which was completely rescued if you added back the deficient protein, suggesting that CRIM2 was sufficient to drive channel activity. Well, in fact, when we looked further, it was the SUMOlation, addition of a small ubiquitin-like modifier to CRIM2 by the enzyme called UBC9, that really had the effect of controlling the channel. And in particular, a graduate student of mine, Eric Destrude, found that it was a particular lysine, 374, that governed the control of 1.7. As you can see, if you knock out this lysine, you see the reduction in current. In collaboration with a group of Dr. Khanna, who solved the crystal structure of CRIM2, we could see that the lysine was the most accessible, and these results were consistent with the idea that this position was key to driving sodium currents. Taking this into the sensory neurons, which are the nociceptive cells, we found that although sodium channels are of many different flavors in these cells, 1,7 is the most predominant in small neurons, we found that if you transfected these neurons with either a wild type or a sumo null, missing the 374 site, you saw that the TTX resistant currents were not affected. You saw that the sensitive currents, which included 1,7, was affected. And if you took out 1,7 with using Protox, you saw there was no difference in that the remaining currents to 1,116 were not affected. So by inference, it was 1,7 that was affected. And not only was the trafficking, the density affected, but also because of trafficking, as you can see in these confocal images, surface trafficking of 1.7 is completely impaired in the presence of a sumo null mutation. We came at this a different way, where we looked at designing a small decoy motif that encompassed the simulation site. And this site gave us an advantage to look at it in to not using genetic approaches. And so we came up with this peptide, coupled it to a TAT sequence to make it cell penetrant. And when we flooded the system in cells, we could see using a denaturing IP that we could block simulation of CRIM2. Here's the control condition. And in the presence of the peptide, you see inhibition of simulation, as you would see with the 374A plasmid. Importantly, when you dialyze this peptide into cells, we saw remarkably an inhibition of sodium currents. And most amazingly, beg your pardon, most amazingly, we saw that in animals with a model of neuropathic pain, for example, the SNI, spared nerve injury, when we injected this peptide, we could see a reversal in these red triangles here, a reversal of both tactile and thermal allodynia. So this was really convincing evidence that this mechanism could be amenable to further studies. Delving deeper into the mechanism, we decided to make a mouse, and using CRISPR-Cas editing, we made a knock-in mouse lacking the simulation site, and very reassuringly, as predicted, the wild-type currents were robust sodium currents were found, but in the sumo null mouse, these currents were dramatically reduced, and this was directly because of a trafficking effect on the channel. When we looked at taking this to pain models, knowing that now the simulation was affected and the trafficking of the channel was affected, we next tested these sumo null mice for the ability to withstand or develop pain. And in the spared nerve injury model, we saw that wild-type animals develop pain, but neither female or male homozygous mice developed as robust pain as the wild type. But more amazingly, they did not have any persistent pain. So all of this data sets up the idea that CRIM2, particularly simulation of CRIM2, is key to the initiation of neuropathic pain and to the development in neuropathic pain. So this was a great time because we understood the mechanism, and we had built a model which shows that UBC9, the enzyme that brings sumo to CRIM2, we had an understanding of that interaction, and we also already knew that we could block the interaction between UBC9 and CRIM2 to prevent simulation of CRIM2 with a decoy peptide. So we leveraged these ideas and these data to come up with the hypothesis that if we could do the same thing with small molecules, we could indirectly regulate the channel in this manner. And so we would inhibit with small molecules this interaction and prevent simulation, and this was converted to a no pain scenario. We began this with a docking effort, and a 50,000 compound screen was performed by Drs. Khanna and Vijay Gokhale, and this resulted in binding of many compounds to this pocket shown here in this 3D crystal structure. And then some of those compounds were screened and then iteratively improved. After about 250 so compounds, we came up with a compound called BPBI or 194. And this is really the compound for which a patent was awarded recently. And this is what I'll talk to you about in the next few minutes. So now, having understood the mechanism that simulation was key, and we had discovered this 194 compound, we tested to see if the compound would work as we predicted. So first, David Scott in Mai's lab showed that compound binds to CRIM2 but not CRIM5 or UBC9. If you bind to this, this would be a problem, and so this is a good thing. It also inhibited the interaction between CRIM2 and UBC9, as shown in this MST experiment. And importantly, taken into cells, again, we were able to recapitulate the inhibition of SUMOylation as we saw earlier with the peptide or with this SUMOylation deficient plasmid control. 194 or BPBI inhibited surface trafficking of the channel as seen in biotinylation experiments here, or in confocal imaging where you can clearly see an annulus of 1.7 on the surface versus in the case with the compound. So it was inhibiting the trafficking. And importantly, because of this, it was inhibiting the currents. So DRG currents here are from normal, from DMSC treated rad DRGs were inhibited when treated with 194. All of this reduction was because of 1.7, because if you added the selective Pfizer compound, PF, shown here, we saw no further inhibition. So everything that was being inhibited was 1.7 selective. And it was only selective 1.7, as can be seen here, as other channels were not affected. Also, this calcium was not affected. DRGs from mouse, pig, and human were also showing similar degree of inhibition with 194. And remarkably, the most convincing proof here comes from the fact that in sumo-null, in DRGs from sumo-null mice, which have this mutation, we saw no inhibition with 194, suggesting and proving that 194 blocks 1.7 specifically via CRIM2 SUMOylation dependent mechanism. So this was really exciting, and this led us to then test this compound, which is our lead compound, in models of neuropathic pain. Here's our two examples. We tested in chemotherapy-induced neuropathic pain as well as the nerve injury model, and as you can see, given orally, this compound reversed neuropathic pain. So it's pretty exciting. So this was an evoked model of pain, but we also sent out this compound, and Dr. Peter Grace from UT MD Anderson's group performed conflict avoidance assays, mechanical conflict avoidance assays, where a rat or a mouse wants to go to the dark side, but it has to cross over a spiked floor. So an animal in pain will not traverse unless it has a drug that's causing pain relief. And so in our compound, BPPI shown in green here, was able to enter this compound to get pain because it had pain relief. And so in this non-evoked assay, we also showed relief of pain with our compound. And we've done a lot of additional studies, but I'll leave you with a couple small ones showing that chronic administration of 194 is very, as most patients will take drugs chronically. So chronic administration of 194 does not cause any tolerance. We did not see any real loss of efficacy during the time course of the experiment where the pump is on. Neither did we see any addictive properties, and this was done in Todd Vander's lab at the University of Arizona, where the compound, the animals didn't prefer any chamber based on compound administration. So with this, I'm going to conclude on 194. I know it was really rapid, and switch to before I end, switch to a very exciting discovery that our lab made a few months ago, which really falls down onto our studying CRIM2 and being in the right place at the right time. So we were studying CRIM2, and I alluded to what we've been doing for the last few minutes or so. And what we found were a couple of papers that showed a new protein called neuropilin. Well, why is that important? Well, neuropilin is a protein that sits upstream of CRIM2. So that caught our attention. Neuropilin was shown to be a receptor protein sitting on the membrane that was able to have SARS-CoV-2, spike protein SARS-CoV-2 bind to it in order to enter it. Well, that got us very interested because we already knew from earlier studies from other people that vascular endothelial growth factor, VEGF, bound to neuropilin to induce pain. So the idea came to us that what if the spike protein SARS-CoV-2 could occlude this particular site? And that's shown in the cartoon here. And what it shows is that neuropilin has a binding site for VEGF, which is exactly the same binding site for SARS-CoV spike protein. So that was fantastic because that got us really interested in doing this. And what we found essentially was if we took animals, added VEGF, we could see pain, but when we added spike protein, we could reverse the pain. And this was through a CRIM2-dependent mechanism, which we're studying, but mostly it was through suppression of currents to sodium and calcium channels and led to decrease in excitability and pain relief properties. So I know I was pretty fast, and my time is almost up. So I just wanted to acknowledge the funding from the National Institutes of Health and the wonderful people that work in the lab. And I'll leave you with this final slide that gets to the heart of what I was trying to convey, which is that there are many potential targets in the peripheral nervous system. We chose one particular target, NAV 1.7, and we think that we understand the mechanism of it. We know how it works. It relieves pain chronic orally. It is very specific to reduction of 1.7. It also, what I didn't show you, it also potentiates and is synergistic with other analgesics like morphine and gabapentin. And finally, it is not addictive. So I apologize for going super fast, but thank you for listening.
Thank you very much, Rajesh, Professor Khanna. That was superb. Thank you so much for that really, really fascinating talk. So we're going to take questions, but we're going to take them at the end of both of our invited talks today. So do you sit tight, and hopefully some questions will come in on the feed. So to anybody listening, if you have a question, just pop it into the question box. If you see the little question mark on your screen, you just tick on that, and then it'll open up a question box and you can type in your question there. If you have a question for Professor Khanna or a question for Professor Finn, it'd be great if you could direct the question if possible. So thank you very much, Rajesh. So I'm now delighted to introduce our second invited speaker today. Our second invited speaker is Professor David Finn. Professor David Finn is a professor and head of pharmacology and therapeutics at the National University of Ireland, Galway, where he is also co-founder and co-director of the Centre for Pain Research. Dave is also currently president of the International Cannabinoid Research Society. So prior to joining NUI Galway, Professor Finn earned a PhD from the University of Bristol, after which he worked as a postdoctoral research fellow at the University of Nottingham. So we are really delighted to have Professor Finn here today to speak at our webinar. Thanks, Professor Finn.
Thanks very much, Ailish. Thank you for organising this and for the invitation to participate, and thanks to the Biochemical Society, Portland Press and Neuronal Signalling for organising this webinar. So I'm going to speak about pain and negative affect with a focus on the endocannabinoid system. And pain and negative affect, by which I mean anxiety, depression and stress, they share a reciprocal bidirectional interaction whereby one can modulate the other. And this is important because we know that there's a high degree of comorbidity between chronic pain, anxiety, and depression. About 50% of patients with chronic pain also suffer from comorbid anxiety or depression. And common pharmacological treatments are used to manage both chronic pain and affective disorders, such as, for example, antidepressants or anxiolytics like pregabalin. And there are also shared neural substrates. We know this from functional neuroimaging and neuroanatomical studies. Brain regions like the anterior cingulate cortex, insular cortex, amygdala are commonly implicated in chronic pain and negative affect. Now, when we look specifically at the relationship between stress and negative and pain, the picture depends on the precise nature and intensity of the stressor in question. So stressors which are robust, intense, fear, and immediate threat to homeostasis tend to suppress pain through the phenomenon of stress-induced analgesia, whereas stressors that are more chronic, moderate, future-oriented tend to do the opposite and exacerbate pain through the phenomenon of stress-induced hyperalgesia. And in my lab, we're interested in both of these phenomena, and I'm going to focus on our work on the role of the endocannabinoid system in both stress-induced analgesia and stress-induced hyperalgesia. So a few years back, we published this review article where we reviewed the neurobiology of stress-induced hyperalgesia, and what was very clear is that it's associated with maladaptive changes throughout the descending inhibitory pain pathway. And so what we see is there are maladaptive changes in a variety of neuronal pathways and receptor signaling systems, such as, for example, the GABAergic and glutamatergic systems, cholecystokinin, corticotrophin releasing factor, and indeed recent evidence for maladaptive alterations in the endogenous cannabinoid system. So just by way of introduction, a little more detail on the endogenous cannabinoid system, or so-called endocannabinoid system. It's a system that's a lipid signaling system comprised of at least 2 receptors that have been discovered and cloned and characterized to date. These are the CB1 and CB2 receptors, and these are both GI-GO protein coupled receptors. The endogenous ligands for these receptors then are the endocannabinoids, and the 2 best characterized here are anandamide and 2-arachidonoglycerol, or 2-AG. And then there's a variety of enzymes that either synthesize or degrade the endocannabinoids. For example, fatty acid amide hydrolase, which preferentially degrades anandamide, and monoacylglycerol lipase, which preferentially degrades 2-AG. And indeed, there are also non-CB1, non-CB2 receptor targets for cannabinoids, including plant cannabinoids that are derived from the cannabis plant, and endogenous cannabinoids. And some of these include the PPARs, TRPV1, and GPR55. Interestingly, the endocannabinoid system signals retrogradely. So endocannabinoids are produced on demand, synthesized on demand postsynaptically, and they are released and then bind to CB1 receptors expressed presynaptically on the presynaptic nerve terminals, where they usually serve to suppress neurotransmitter release and neuronal activity. So it happens that you find the components of this endocannabinoid system distributed throughout the descending pain pathway in all these regions that are so important in pain regulation and also negative affect, stress, fear, anxiety. Regions like the prefrontal cortex, amygdala, periaqueductal gray. And when this pathway is activated, it generally tends to suppress nociceptive transmission. So we started out by asking, is there a role for the endocannabinoid system in pain hyperresponsivity, so exacerbation of pain in negative affective states like anxiety and depression? And to do this, we're taking both preclinical and clinical approaches. I'm going to focus mostly on our preclinical work in animal models. And here we're using an inbred rat strain called the Worcester-Kyoto rat, which has an anxiodepressive phenotype. So in an open field maze, in an elevated plus maze, the forced swim test, these animals, this Worcester-Kyoto genotype has an high anxiety, high depressive-like behavioral state compared to Sprague-Dawley rats, which are the most commonly used comparator strain. And in tests of pain, such as the hot plate test and formalin test, these Worcester-Kyoto rats show a heightened nociceptive response, heightened nociceptive behavioral response to both heat pain and to inflammatory pain with formalin injection. So we use them, if you like, as a model of heightened pain responding against a negative affect background, a high anxiety, high depressive background. And these Worcester-Kyoto rats have been studied in terms of the endocannabinoid system for depressive-like behavior. And what's clear is from this paper published by Vinod and colleagues a few years ago is that a dysfunction in Fa, the enzyme that preferentially degrades anandamide, is associated with depressive-like behavior in the Worcester-Kyoto rats. And if you chronically treat these Worcester-Kyoto rats with a Fa inhibitor, it has an antidepressant-like effect. So in some of our earlier work published in Pain, we were able to demonstrate that impaired endocannabinoid signaling in the brainstem, in the rostroventromedial medulla, the RVM, underpins this heightened pain responding in the Worcester-Kyoto rat strain. But of course, the RVM in the brainstem receives innervation from the midbrain periaqueductal gray, PAG. And so we next turned our attention in more recent work to the role of the endocannabinoid system in the PAG. And here I'm showing you some results. We did a very comprehensive characterization of the endocannabinoid system in the PAG of Worcester-Kyoto and Sprague-Dawley rats. But just to show you a selection of the more interesting data, we found high levels of the endocannabinoid 2-AG in the lateral PAG of Worcester-Kyoto rats compared to Sprague-Dawley rats, significantly higher levels, and significantly lower levels of the CB1 receptor in the lateral PAG of the Worcester-Kyoto rats compared to the Sprague-Dawley comparator strain. So we hypothesized that these alterations in the endocannabinoid system, or differences in the endocannabinoid system between the Worcester-Kyoto and the Sprague-Dawley rats, may underlie the heightened pain responding in that strain. And so to test that hypothesis, we next carried out some pharmacological challenge studies where we injected CB1 receptor agonist ACEA directly into the lateral PAG of both strains. So in Sprague-Dawley rats, we see that this CB1 agonist gives us a nice anti-nociceptive effect, significantly reducing formalin-evoked nociceptive behavior. And that effect is blocked by co-administration of a CB1 receptor antagonist, AM251. But if we do the same experiment in Wistar-Kyoto rats, we don't see these effects. We see no anti-nociceptive effect of CB1 receptor agonist administration into the lateral PAG of the Wistar-Kyoto rats. They seem to be hypo-responsive to cannabinoid receptor agonism in this region, the PAG. And if we look post-mortem at CFOS expression as a marker of neuronal activity, here we see in the Sprague-Dawley rats that the CB1 receptor agonist increases FOS expression in the brainstem and reduces FOS expression in the dorsal horn of the spinal cord. So this is consistent with the idea that it's engaging the descending inhibitory pain pathway in Sprague-Dawley rats, but we just don't see these effects in Wistar-Kyoto rats. So they're, again, hypo-responsive in terms of recruitment of the descending inhibitory pain pathway to cannabinoid receptor agonism. And in terms of the circuitry of the RVM, it's quite interesting. It has these on cells and off cells. The on cells are thought to facilitate pain responding, and the off cells are thought to dampen down pain responding. And so in collaboration with Dino Maioni and colleagues in the University of Naples, we recorded from the on and off cells of the Wistar-Kyoto and Sprague-Dawley rats at baseline and in response to mechanical stimulation. And the strains are markedly different. So in brief, the Wistar-Kyoto rats have exacerbated on-cell responding and reduced off-cell responding, which again is consistent with the idea that they have a heightened pain response and a dysfunction in the descending inhibitory pain pathway. So just to summarize the results that presented so far, the Wistar-Kyoto rat strain is a anxiodepressive, high anxiety, high depression related behavior strain. We see hyperalgesic response to formalin injection into the hind paw, a blunted endocannabinoid signaling in the RVM, differences between the endocannabinoid system and the lateral PAG of Wistar-Kyoto and Sprague-Dawley rats, and differential responsivity to CB1 receptor challenge and agonism between the two strains. And you can see that at the level of nociceptive behavior, at the level of CFOS expression, and indeed electrophysiologically as well. So we feel that dysfunction of this endocannabinoid system in the PAG-RVM circuitry likely contributes to heightened pain responding in the Wistar-Kyoto genotype, which is associated with negative affect. Okay, so so far I've been talking about how negative affect and fear, stress, anxiety, how they exacerbate pain, and I'd next like to talk about how the opposite to that, so how fear and stress can suppress pain. And to study this, we use a paradigm which we call fear-induced analgesia or fear-conditioned analgesia. And this is pain suppression in response to learned Pavlovian conditioned fear. It's a very, very potent form of endogenous analgesia. And so therefore it's of interest to us because if we could understand the neurobiology and physiology of this endogenous analgesic response, well, it would be interesting, of course, from a fundamental physiological perspective, but also it may give us some insights into novel therapeutic targets for pain. Fear-conditioned analgesia is also expressed in rodents and humans, so it has some translational value. And it's mediated by activation of the descending inhibitory pain pathway. So that pathway that we looked at earlier, when it's activated, it is critically important in mediating fear-conditioned analgesia. Now, to model this in our laboratory, we use a two-day paradigm. On day one, the rats undergo fear conditioning to context, Perspex arena. And on day two, they are injected with formalin into the hind paw and then re-exposed to the arena. And re-exposure to the arena results in a suppression of formalin-evoked nociceptive behavior through fear-conditioned analgesia. And we can interrogate the neurobiology of this response by systemic or intracerebral injection of drugs that modulate the endocannabinoid system. So in initial work with this model, we could show that if you block CB1 receptors pharmacologically with the antagonist rimonabant, that this completely prevents fear conditioned analgesia in these rats. So we have our fear conditioned rats, non-fear conditioned rats. The vehicle treated rats express robust fear conditioned analgesia, but the rats receiving the CB1 receptor antagonist rimonabant do not. And you can see this at the behavioral level, but you can also see it at the level of immediate early gene expression as a marker of neuronal activity in the dorsal horn of the spinal cord too. We next administered a drug that would prevent the breakdown of endocannabinoids. So this is the drug URB597. It's a Fa inhibitor. And that does the opposite. So that enhances fear conditioned analgesia. And again, this is consistent with the idea that during fear, endocannabinoids are being synthesized and released on demand. They're binding to CB1 receptors, and they're facilitating this endogenous pain suppression. And in a series of papers over the last 10 years or so, which I don't really have time to go into in detail, we've been interrogating the neural substrates and brain regions that are important in this endocannabinoid-mediated fear conditioned analgesia. And some of those papers just summarized here, and we've shown a key role for the dorsolateral periaqueductal gray, for the basolateral amygdala, for the prefrontal cortex, and ventral hippocampus also. And here's just a selection of some of those data, and what we show is that fear conditioned analgesia is associated with increases in endocannabinoids and anandamide in the basolateral amygdala and dorsolateral PAG. And if we inject CB1 antagonists directly into these brain regions, we can prevent the expression of fear conditioned analgesia. Okay, just as I move into the latter part of the talk, I've been talking to you so far about how anxiety and fear and negative affect modulate pain, but I'd also like to turn it around now and look at how pain can modulate negative affect, because this is also very important clinically. And some nice work from Rafael Maldonado and colleagues in Barcelona a few years ago demonstrated that indeed there's a role for the endocannabinoid system in the emotional manifestations of osteoarthritis pain in animals. And we asked whether there might be a role for the endocannabinoid system in emotional manifestations of neuropathic pain using a rat model of spinal nerve ligation, which is a peripheral nerve injury model of neuropathic pain. And here again, we have our two strains, the Worcester-Kyoto and the Sprague-Dawley rats. And we're looking here at the forced swim test, which is a test for depressive-like behavior and antidepressant-like activity. And we're looking here about 38 days following peripheral nerve injury in both strains. And so what we have, the sham animals that do not have the peripheral nerve injury, and the spinal nerve ligation animals that do have this neuropathic pain-like state. And so what you see is that the Worcester-Kyoto sham animals have greater immobility in the forced swim test compared to the Sprague-Dawley shams, and this is entirely expected. And I've told you already in the talk that Worcester-Kyoto rats have a high depressive-like behavioral phenotype, and so that makes sense. And then when we layer on the spinal nerve ligation, then the peripheral nerve injury, we see an augmentation of this depressive-like behavior in the Worcester-Kyoto rats, but not in the Sprague-Dawley rats. So we followed up this post-mortem work. We actually showed that this augmentation is associated with increased CB1 receptor expression in the basolateral amygdala, which gave us a clue that maybe the endocannabinoid system could be an interesting target here. And then we carried out a subsequent experiment where we chronically treated the nerve-injured Worcester-Kyoto rats with a Fa inhibitor, URB597, for about 5 weeks. And we're able to show that this had an antidepressant-like effect. It's a modest effect, but statistically significant reduction in the immobility in the forced swim test in these nerve-injured Worcester-Kyoto rats compared to control rats receiving vehicle rather than the Fa inhibitor. So that's all well and good and some interesting data in animal models, but what's the story clinically, and what does the picture look like in human patients and for cannabinoids? And I guess we've reviewed the literature here in a few different articles in recent years. It's just a table that I've pulled out from one of our reviews, which summarizes the literature on cannabinoids for treatment of comorbid pain and anxiety, depression. And for the most part, what you see is that the cannabinoids that have been studied do indeed reduce pain and also the comorbid anxiety, fear, depression. But what I would say is that there's a very limited range of cannabinoids that have been studied to date, largely only THC or THC analogs for the most part. So we do need more studies with different types of cannabinoid drugs, with FAH inhibitors, MAG lipase inhibitors, to advance this field. There is some interesting work looking at human SNPs, polymorphisms in the FAH gene, which have been linked to differential pain responding and differential anxiety responding. And this paper up here in the top left, particularly interesting, got a fair bit of media attention last year. It's a lady in Aberdeen in Scotland who has this FAST SNP who has very, very high levels of anandamide and never feels pain and is always happy and never anxious. And she had surgery and didn't feel any pain whatsoever post-surgery. So that's very interesting, and I think further proof of concept that FAH, it represents an interesting target for chronic pain. Fear condition analgesia is mediated by the endocannabinoid system in these brain regions shown here. We also see some evidence, in fact, for reduced expression of fear condition analgesia in Wistar-Kyoto rats, which I haven't shown you, but we have found that, and that deficits in FAST substrates, including endocannabinoids, may underlie nerve injury-induced depressive-like behavior in Wistar-Kyoto rats. The final bit of data I just want to present to you for the last minute or two is in the area of post-operative pain, and this is an animal model, a rat model of post-operative pain associated with inguinal hernia repair, groin hernia repair. We developed this model a few years ago and published it, and here's work showing that if we administer FAH inhibitors, either a peripherally restricted FAH inhibitor or a centrally acting FAH inhibitor, then these have antinociceptive effects in this rat model of post-operative pain. And finally, we've been doing some clinical research in women undergoing cesarean section. C-section is associated with certainly acute postoperative pain and in a significant proportion of women, chronic postoperative pain too. And we've been measuring the endocannabinoid levels, circulating endocannabinoid levels in blood, and they're markedly changed post cesarean section compared to pre-cesarean section. Increases in 2-AG, reductions in anandamide, OEA, and PEA. And so we're interested in this in the context of endocannabinoids being potential biomarkers for predicting postoperative pain. So to summarize, the endocannabinoid system is an important lipid signaling system that I think plays a key role in regulating this balance between negative affect, fear, anxiety, depression, and pain. This is a very complex balance, and it is going to depend on many different factors, the levels and types of endocannabinoids, the precise receptors they act at, and of course the brain regions and circuitry. But hopefully our data may be of some relevance to the development of endocannabinoid-based therapeutics where you have comorbid anxiety, pain, and depression. I'd just like to thank all the people in my group over the years who've contributed to this work. Some of the postdocs and PhD students who've contributed to the work I presented are highlighted in bold there, and collaborators in Galway, particularly Michelle Roach here in physiology in Galway, other collaborators in Galway, London, Naples, and of course the funding agencies that have supported the work. And maybe just while I have the floor, just to draw your attention to this cannabinoid meeting. It's the International Cannabinoid Research Society Symposium, which we hope to host here in Galway in 2022. Fingers crossed, COVID permitting. And if you're interested in this area, we'd love to welcome you to Galway for that. Thank you.
Thank you very much, Professor Finn. Fascinating run through the very complex relationship between affect, pain, and where the cannabinoids fit in, or the endocannabinoid system fits in. So we're going to head into the Q&A part of the webinar. Just to remind all of our participants that are watching at home or at work, if you have a question, just to hit the question mark symbol on your screen, and you can pop a question in there, and you can direct it to Professor Khan or to Professor Finn. That would be great if you could do that. So we have some questions in. So the first couple are for Rajesh. So Joseph Parsons has a question about, if you are to deliver your decoy peptide as a therapeutic for humans, what would the mechanism of delivery be, because of issues related to, undergoing modifications or digestion, depending on the route of administration?
Well, thank you very much for that question. So partly for that very reason, with the hurdles with using peptides, we had resorted to developing small molecules. So really pushing forward the idea that small molecules can recapitulate the key findings that we saw with the peptide. The peptide and what we were doing with that was really a proof of concept to set up the idea. However, that said, the peptide works beautifully, as you saw, with some other findings I didn't share with you. It's not addictive, it doesn't have any off-target effects. And currently, at least in the animals, we had done intrathecal delivery. We had done in the past oral delivery as well, and somehow the peptide was spared from degradation. So it is possible, if we were to go ahead with developing the peptide further, we could encapsulate it perhaps, or stable the peptide. We haven't figured out how to deliver it beyond the initial studies, but we've been playing with ideas of encapsulation, maybe perhaps a nanoparticle-based conjugation to it. But again, the whole idea was to take that as proof of concept and then guide us to development of compounds.
Okay, there's another one here for you, Rajesh, from Vivian Tawfik. Do you anticipate that certain types of pain will be particularly sensitive to your compound?
Thank you, Vivian. That's a great question. That's something that we've been thinking for a long time. As you know, NAV 1.7 in particular, the target here, is important for a variety of pain, at least what's been seen in preclinical models and in humans as well. What you also know is that 1.7 targeted compounds have really not fared well, and they've been tried for post-herpetic neuralgia, for example, for dialysis-induced itch, and so because 1.7 also controls itch. So at this point, for us, with our lead compound and improved versions of that, what we're trying to do is to go after chemotherapy-induced peripheral neuropathy because it's so prevalent, and we have clear evidence from our work and from work across the globe from pain labs that this is going to be potentially effective there. So I would say, although the strategy that we're pursuing, which is this allosteric indirect regulation, is likely to be broadly relevant across these pain models, I would say for us, going forward, at least for the development perspective, we would go with chemotherapy-induced peripheral neuropathy.
Great, thank you. One for David. David, somebody wants to know, Pierre Depping would like to know, can you tell us a little bit more how the lipid signaling in the endocannabinoid system works?
Was that how lipid signaling works?
Yeah, how the endocannabinoid system works as lipid signals.
Yeah, sure. So endocannabinoids are lipids essentially. They're derived from arachidonic acid. So they are lipid-like molecules, highly lipophilic, and the signaling works in a sort of on-demand manner. So endocannabinoids are not stored in vesicles. So they're not like classical neurotransmitters, which are stored and released by exocytosis. Endocannabinoids are synthesized on demand in response to stimuli, often physiological stimuli. So they'll be synthesized in response to pain, for example, or stress, or other physiological stimuli, and they're made from phospholipid precursors. They're made that's facilitated and catalyzed by enzymes. So for anandamide, it's NAPLD largely, and for 2-AG, it's DAG lipase. And so they're synthesized on demand, released. They easily, traverse membranes, diffuse out into the synapse, and then they serve to modulate neuronal function. So they're sort of like neuromodulators. It's a neuromodulatory system, and particularly important in modulating, for example, GABAergic neurotransmission and glutamatergic neurotransmission. So you find a high expression of CB1 receptors on GABAergic interneurons. So yeah, in that sense, it's a lipid signaling system because the endocannabinoids themselves are essentially lipids.
Yeah, so they're quite different to a lot of the more classical neurotransmitters in our brains. There's another one for you.
Absolutely.
So the FASNP that was found in the patient who was pain insensitive, has that been subsequently found in other patients? Was that a
Not with such dramatic results. I don't think. There are a variety of different SNPs, and there have been other fast SNPs reported, and in some of the papers I showed there on that slide linked fast SNP, for example, to postoperative pain, and and also to anxiety and fear extinction as well. But that particular SNP was I think the first report of that, as it was a very interesting. There was two polymorphism, two genetic alterations in that one person, I believe, and there was the Fa protein was deficient, and it was almost like a decoy Fa in there as well, which was encoded for by a SNP. So that was the interesting thing there, and the ultimate the end result for the person was that they had very low levels of Fa activity and therefore very high levels of anandamide, which then conferred analgesia and anxiolysis, you know. So yeah, there are other SNPs that have been reported, but not to my knowledge that particular combination that was found and reported in that lady from Aberdeen.
I wonder, do these people also have issues with weight regulation just because of the role of the endocannabinoid system in driving appetite? You know?
Yeah, I don't think that was a reported or a feature there in that particular case. But definitely, CB1 plays an important role in appetite regulation, and of course, we all know about cannabis and the munchies, I guess, and also that the rimonabant, which is one of the drugs I presented some data for, CB1 antagonist or inverse agonist, that was on the market a few years ago for the treatment of obesity, actually, and so, you know, blockade of CB1 receptors does appear to have an effect on, you know, appetite regulation and weight gain. But the flip side to that is the drug had to be taken off the market because of suicidal ideation and tendency towards depression, which I think speaks to the role of the endocannabinoid system in negative affect and in mood regulation.
Yeah, it's fascinating story of rimonabant and obesity. Back to you, we have one here about targeting CRMP2. This comes in from Harrison Stratton. Has your group investigated other residues of CRMP2 that could potentially influence chronic pain?
Yes, we have. Thank you for that question. So it's this protein CRIMP2 CRMP2 was initially identified in the context of its ability to guide and grow axons by binding to tubulin. And so our work has really sort of diverted its canonical role now to seeing whether it's available for conversations or dialogue with channels to affect their activity. And in these studies, we discovered that there is indeed sort of a code built into the protein where different modifications drive it towards different pathways. So for example, loss of simulation, which I talked about today, was sufficient to convey regulation of 1.7. In other studies, we found that phosphorylation, in particular residues within CRIM2, can drive its interaction and activity regulation of calcium channels. There are reports from our work and others that show interaction with an NMDA receptor. So it's a multifunctional protein, and coded into the structure and the sequence of CRIM2 are these modification sites, which are really a code for driving and having different activities. So we are looking at different residues that drive either phosphorylation, simulation, and some other modifications. And ultimately, it's going to be sort of an interplay between these residues and these modifications that will determine the outcome for chronic pain.
Great. I have another question here from Ryochi Taguchi from the Philippines. Hi, Ryochi. It's nice to know there's people that far away listening to us today. So this one is also for Rajesh. It's about a preprint publication about the 1.7 channel from McDonald et al. In 2020. I don't know if you're aware of it, but it says that it printably modulates the opioidergic system. And he's wondering if your inhibitory compounds might also affect this system.
I was hoping to avoid that question, and absolutely great question. Thank you, and hello, This is a great venue. We can talk to people across, amazing, across the world. So thank you for that question. Absolutely, to answer your question in a short manner, absolutely, our mechanism is directly affecting the opioid system. So for those that don't know, I'll give a little bit of context. In the last 5 to 10 years, particularly in groups in the UK, John Wood and colleagues have shown that the reason that the sodium channel targeting drugs are failing is because they're not engaging the opioid system. So what his group has shown particularly is that you have to completely suppress 1,7, which then drives upregulation of pro-pre-enkephalins and some of these endogenous opioids. And that could explain why the compounds that have been tried by other folks and other strategies haven't worked. So we've looked at that mechanism and we have data. All of that data, by the way, I showed was unpublished with 194. So we do have evidence that our compound, through the suppression of CRIM2 stimulation, does engage and upregulate, much like John Wood's group showed, the pro-pre-enkephalin pathway. And we have some behavioral evidence to suggest that indeed there is a connection that is through the opioid pathway. This was also verified by a group, Tim Huscho's group in Aachen, Germany. So we are very convinced now that we are indeed engaging the mechanism, and therefore, perhaps our compound is likely to succeed where others have failed. Great question. Thank you.
Great. Thanks, Rajesh. There's another question here from Loretta Branley, and unless there's another Loretta Branley somewhere in the world, she's one of our own students here in Galway. So this one is actually for Rajesh. So Loretta wants to know, Rajesh, if damage to neurons has been known to lead to overexpression of sodium channels. So she wants to know if there was a differential overexpression in the 1.4 and 1.7 channel in your models compared to other sodium channels.
There is. That's been reported. Thank you for that question as well. That has been reported by lots of groups. We also see an upregulation. But curiously, and this I failed to mention this within the short time frame, the reason we're going after CRIM2 stimulation as a potential way to manipulate 1,7 is because in the context of chronic pain, in the injury situation, the level of CRIM2 stimulation goes up. So it's almost like a biomarker. And in all cases, we've seen the level of CRIM2 stimulation goes up, the channel expression goes up. So the targetable proteins are themselves plastic. They're showing this change after injury, and which really then reinforces the fact that we can control them in this manner. But yes, we are seeing upregulation. We haven't followed it across a time course, but we do see upregulation both at the current density level and the protein level in the channels, and particularly in CRIM2 stimulation. And the total CRIM2 is not changed, but the stimulation has changed.
Okay, brilliant. There's one here for Dave. Really interesting question. So it's from Megan Hannon, and she asks about epilepsy. So she said that there's a high degree of comorbidity with anxiety and affective disorders and epilepsy. So she's wondering if there's been any studies showing that there's been improvement in affective states in epileptic patients that are on endocannabinoid modulating drugs.
Oh yeah, that's a very interesting question. Well, the big development in epilepsy, I suppose, in recent years in the context of cannabinoids has been the launch to market of Epidiolex, which is CBD or cannabidiol, drug by GW Pharmaceuticals. Got FDA approval there a couple of years ago, and this is specifically indicated for childhood epilepsies, so Dravet syndrome and Lennox-Gastaut. You know, it hasn't been on the market very long. The clinical trials were quite positive, and of course that's why it got its FDA approval. But I'm not so aware of data on anxiety and depression in that cohort. You know, I mean, the primary endpoints there, of course, were seizures, and it is effective, cannabidiol, in terms of effects on reducing seizures. But I think we do need more data on some of the comorbid symptoms of epilepsy in those kids, and also, in fact, in adults with epilepsy as well. Yeah, it's a really interesting question. I just don't think we have the data. I'm not aware of the data, at least for CBD and negative affect in epilepsy. There are reasons to think. I mean, CBD is very different to FA inhibitors, for example. CBD is has quite a complex pharmacology. It's probably a negative allosteric modulator at CB1, but it may also have effects at GPR55 and other targets as well. Whereas obviously FAH inhibitors are elevating, endocannabinoids and anandamides specifically, and then acetylethanolamines, and largely, then having effects through the targets that anandamide binds to, CB1, CB2, and others. So, yeah, it's hard to make a direct comparison with the kinds of drugs I was talking about and in my talk and CBD. But yeah, the whole question of anxiety, depression, comorbidity, and epilepsy, and whether there's a role for the endocannabinoid system, I think it needs further study.
Yeah, great. Who have we got? We've got one here from Yufeng Z for Rajesh. What do you think is the relationship between CRIMP2 or CRMP2 and 1.8 channel?
Thank you for that question. So we don't know. The short answer is we don't know. We know that CRIMP2 is able to have regulation of on 1.7 and on CAV2.2, for example, and we see some residual currents still left even after the simulation is inhibited that are not 1.7. So there's something that's still, as you saw from the data and some of our papers, that there is some residual currents that might be still in play. Whether and how they talk to and are regulated by CRIMP2, we don't know. We have evidence, for example, that some of the other currents are not affected by CRIMP2, particularly T-type calcium channels. So we haven't looked at 1.8, but to be honest, if it didn't affect 1.8, it wouldn't be that bad. It is also a high-value pain target. So while I don't have an answer to your question, an unintended for CRIMP2 affecting this channel would not be a bad thing. Thank you.
Okay, and we're coming up to the end of time. I'll take one more question here from Andrew Scarborough. It's for Dave. So Dave, it's in relation to somebody with a chronic pain. Obviously, would have to may have to take medication for long periods of time. He's just wondering about side effects of cannabis-based medicines, and particularly, on the liver, on liver function.
Oh yeah, another good question. Yes. I mean, continuing on with CBD then and cannabidiol, there are some data to suggest that CBD may have some negative effects on liver enzymes, on cytochrome P450 family of enzymes, and indeed reports that in those children who were taking CBD for childhood epilepsy, that levels of some of the other anti-epileptic drugs that they were on were higher when they were taking CBD, probably because of pharmacokinetic effects of CBD, and effects on liver enzymes. So I think, CBD, people often say it's very safe, it's not psychoactive. I think we need to be careful with that. It actually is psychoactive. It's not psychotomimetic in the same way that THC is, but actually it has effects in the brain, and anxiolytic effects, and clearly anti-epileptic effects, and so it must be functioning in the brain, and it does have a degree of psychoactivity. Cannabis more generally, I mean, we could talk about this for a long time. There are obviously risks with chronic cannabis use, in the general population of users who use it recreationally, there's about a 9% dependency to cannabis, particularly if it starts in adolescence and with heavy THC, high THC containing strains. But then there are also risks around, mild cognitive impairment, perhaps, and then peripheral, cardiovascular risks as well. THC can induce tachycardia, can be quite marked. So there are risks, and I think, we probably need more clinical trials with larger, sample sizes for longer duration, particularly in pain. We do have trials. There's about 35 to 40 RCTs. A number of them do show some efficacy, albeit sometimes quite modest. The evidence is probably best for neuropathic pain. But yeah, I think we just need maybe studies of longer duration, larger sample size, and that focus a bit more on adverse effects as well.
Okay, that was fantastic. I really want to thank everybody who participated today, especially Professor Finn from Galway in Ireland and Professor Canna from Tucson in the United States. And to all of you who joined up to this webinar, there was a lot of people listening in today, so we're really pleased about that. So thank you so much for joining in, especially for everybody who sent in a question as well. Thanks to the Biochemical Society and Portland Press for this webinar series, and to neuronal signaling in particular for this particular one on the neurobiology of pain. So before I finish up, I don't know if Dave and Rajesh want to just say goodbye.
Thank you very much for the opportunity. Bye-bye.
Okay.
Thanks again, Eilish, for organising. Thank you to Sandra for her help, and to everybody for joining.
Thank you, thank you. So the Biochemical Society and Portland Press welcome suggestions from anybody for future webinars. So if you have an idea or a topic or anything you want to find featured, just have a look at the website, biochemistry.org/webinars, and find it there. So the next webinar that we will be hosting will be on the 9th of November at 2 o'clock GMT, and it's entitled Inflammasomes and Plant Resistome. And this will be chaired by Gloria Lopez Castellon, along with speakers Jack Green and from the University of Manchester and Cian Duggan from Imperial College London, who are both ECRs, and they're going to share their work with us all. So that's it for today. It's after 4, so we'll sign up. Thank you very much. And thank you very much, everybody. Thanks, Biochemical Society. Thanks, Portland Press. Okay, bye. Bye-bye.